Okay, sorry about the abrupt ending to that previous video. I didn't, again, foresee the fire alarm going off. Right, okay, so we were discussing what the processing of the products of digestion which occurs within the smooth endoplasmic reticulum of these enterocytes. Okay, so we've seen that 3 fatty acids and 2 monoacyl glycerols are going to be reassembled into triacyl glycerols. We've seen that 3 fatty acids and lysophosphoglyscerolipids are going to be reassembled into phosphoglycerolipids. You're also going to get cholesterol molecules being resynthesized into cholesterol esters. Okay, now what these enterocytes are then going to create are chylomicrons, basically, which are a way of transporting large quantities of lipids around the body. Okay, so what we're going to create is chylomicrons, and these chylomicrons are then going to be put into the central lacteal of the villus, basically. So they don't go into the blood, they go into the lacteals. Okay, so let's discuss the structure then of a chylomicron. So basically, chylomicrons have a spherical structure. Okay, so they're effectively a massive great ball of lipid molecules. Now, the outer surface of the chylomicron is made up of a monolayer of phospholipids. Okay, so this is a phospholipid monolayer. Okay, now earlier when we were discussing the difference between phosphoglycerolipids and phospholipids, I promised to give you examples of molecules which are phospholipids but which are not phosphoglycerolipids. So, in order to be counted as a phospholipid, all you actually have to have is you have to have a fatty acid or a long-chain carboxylic acid within your structure. Okay, so you have to have a really long-chain hydrophobic carboxylic acid within your structure. And you also have to have a phosphate group. That's all you have to have in order to be considered a phospholipid. So that's why these phosphoglycerolipids here are considered phospholipids, because they have 2 fatty acids within their structure, and they also have a phosphate group in their structure. Okay, but there are other types of lipids which satisfy these conditions and which are not phosphoglycerolipids, okay, so they don't have glycerol within their structure. So, for instance, other examples of phospholipids are sphingomyelins, okay. So there are certain types of lipid molecule called sphingomyelins, and basically these are structures which have sphingosine as their backbone. So rather than glycerol, they have a molecule called sphingosine as their backbone. They then have a long-chain carboxylic acid stuck off the side of them, okay, via actually an amide bond, and then they have a phosphate group attached to their head up here, like so, okay, and then also of the phosphate group they can have some interesting groups such as a choline molecule or an ethanol-amine molecule, just like the phosphoglycerolipids as well, so we can have some interesting group off here. So basically, not only do they have this long-chain carboxylic acid attached to them, shown in orange here, but they also have this sphingosine molecule, and sphingosine has a very long hydrophobic tail, so in fact they actually have a very similar structure to these phosphoglycerolipids. They have 2 long-chain hydrophobic structures, then a phosphate group, and then some additional potentially polar head here. So that is why when people generally denote phospholipids, they generally draw them like so. Okay, so they draw the 2 long chain tails, okay, because both phosphoglycerolipids and also these sphingomyelins here, they both have these 2 long tails, and then they also draw this polar head here, which represents the phosphate group with the additional structure added on that you have in both the case of the phosphoglycerolipids and also these sphingomyelins. So I'll now use this notation and I'll put these phospholipids like so. Okay, so the polar head of the phospholipid molecule basically faces out to the external world, and this is helpful because these can interact nice and favourably with water. Then the long hydrophobic tails then face into the centre of the chylomicron. Okay, and these can interact with the lipid contents that is going to be at the centre of the chylomicron. Now, I'm going to leave some space up here because we're going to also have proteins in this monolayer. So this outer layer that we have of phospholipids, and you'll notice that it's not like a cell membrane. In a cell membrane you have a bilayer of phospholipids. In chylomicrons you just have a monolayer. So this is a phospholipid monolayer. Okay, now in the centre of the chylomicron you would put all of the lipid molecules which then do not have a polar group. So for instance, you will put the triacylglycerol molecules in here. You will put some free fatty acid molecules. So not all of the free fatty acid molecules that you absorb will be reassembled into triacylglycerol molecules. Some of them will end up in the chylomicrons on their own, okay, as free fatty acids. So here is our triacylglycerol molecule, okay, like so. You might also have some long-chain carboxylic acids which are free. Okay. Other things that you might have are cholesterol esters. Okay, so remember what cholesterol esters look like. So I'll just draw the structure of a cholesterol ester again. So remember, they have this steroid structure where they have these 4 carbon rings which are all next to one another. Okay, like so. Then you have the structure of cholesterol, so we have these methyl groups here, this 7 carbon side chain here with then the methyl group off here, and now we have this alcohol group here which is involved in an ester link to a long chain carboxylic acid here. Okay, when you have cholesterol involved in an ester like this, it no longer has a single polar group. Okay, so before, when you had the alcohol group here, that gave cholesterol at least one shot of having a polar group because the alcohol group was polar. But now that you've attached a long chain carboxylic acid to this alcohol group via an ester link, the whole molecule is hopeless basically. It's extremely hydrophobic. So cholesterol esters will be in the center of the chylomicron basically because they can't interact with water. They're hopeless at interacting with water. Okay, whereas cholesterol, where it doesn't have an ester, okay, so free cholesterol molecules, those actually do have an alcohol group which is slightly polar, so they might integrate into the phospholipid monolayer where their alcohol group is going to face the outside basically. So, in these chylomicrons, you will have these phosphoglycerolipids. You might have a few sphingomyelins making up the phospholipid monolayer. You might have a few cholesterol-free molecules that are in the phospholipid monolayer with their alcohol groups facing towards the outside. And then in the center you have the really hydrophobic molecules, the cholesterol esters, the long-chain free carboxylic acids, and the triacylglycerol molecules. Okay, and that's basically the structure of a chylomicron. Now, these enterocytes here will manufacture these chylomicrons, and in fact I've missed off a key structure. We've shown the lipid molecules, but in fact they have special proteins that are also present within these phospholipid monolayers, and this was why I left this gap here. Okay, so in the phospholipid monolayer portion of the chylomicron, you also have special proteins which are called apolipoproteins. So let me just complete the phospholipid monolayer. So this red blob here is meant to represent an apolipoprotein. Okay, so in the phospholipid monolayer that surrounds a chylomicron, you have certain proteins basically just sitting within the phospholipid monolayer, and these are important with targeting the chylomicrons to certain sites after we have put them into the blood. Okay, so you have a number of different types of apolipoproteins that are all in the surfaces of chylomicrons. Okay, so the apolipoproteins which you find within the phospholipid monolayer of chylomicrons are apolipoprotein B48. And that's a very important one. That's the main one that you find in the outer surface of chylomicrons. Okay, so the notation for apolipoproteins is just to put apo and then the name B48. But you should read it apolipoprotein B48. Another one which is present within chylomicrons is apolipoprotein C2. Also we have apolipoprotein C3. And then finally, you also have apolipoprotein E present within the phospholipid monolayer of chylomicrons. So all of these apolipoproteins you will find in the outer surface of these chylomicrons. And as I say, they're important for targeting the chylomicrons to certain portions of the body after we've actually put them into the blood. Okay, so these enterocytes will be producing these chylomicrons, which are an example of a lipoprotein. And basically a lipoprotein is just this exact structure that I've shown you here. It's a phospholipid monolayer with apolipoproteins within them. And then in the center you have the really hydrophobic lipid molecules which can't interact with water at all and therefore have to be shielded from water by the phospholipid monolayer here. So they're called lipoproteins because they have a protein component which are the apolipoproteins, and then they also have a lipid component. And this is basically the major way that we transport lipid molecules around the blood, because obviously they can't actually dissolve in the blood on their own. Okay, right, so these enterocytes will be making these chylomicrons, and basically these are absolutely massive in the world of lipoproteins. Most lipoproteins are not as big as these chylomicrons. These are giants, okay, so they're big compared to other forms of lipoproteins, such as LDL, HDL, VLDL, things like that. Okay, right, so what's now going to happen? Well, basically the enterocytes are going to release these chylomicrons into the lacteals. Okay, so it will go into the lacteal, not the blood. They release them into the lacteals. So they go in the lymphatic blood vessels, basically. So now what we need to talk about is where do all of the lymphatic blood vessels of the intestine drain into? And now it's time for a bit of anatomy. Okay, right, so basically if we look at the lumbar vertebrae, okay, so remember the vertebral column consists of loads and loads of vertebrae stacked on top of one another, and all of these vertebrae have names. So the ones up in the neck are named the cervical vertebrae, the ones in the thoracic region are named the thoracic vertebrae, the ones down at the base of the abdomen are named the lumbar vertebrae. Okay, so basically we're going to look at the top 3 lumbar vertebrae. Okay, and here are their bodies, okay, the vertebral bodies. So we have L1 here, L2 underneath it, L3 underneath that. So this is the top up here, and this is the bottom. This is going down towards your feet, this is going up towards your head. Okay, so basically, lying on the front of the L2 vertebrae, you have a huge, great sac of lymph, basically. And this great big sac of lymph is known as the cisterna chyli. Okay, so cisterna chyli is basically a huge, great sac of lymph. And basically it has 2 huge trunks coming into it. Okay, and these come from the 2 lower limbs. So these 2 big lymphatic vessels that drain into this huge, great sac here are known as the lumbar trunks. Okay, so this is the lumbar trunks. So you have a left lumbar trunk here and a right lumbar trunk here. Of course, we're talking about patient's left and patient's right. Okay, and the left lumbar trunk will drain the left lower limb, and the right lumbar trunk will drain the right lower limb. Now, basically, all of the lymph that comes from the intestines basically drains into this cisterna chyli here. Okay, And then what's going to happen is the cisterna chyli then has a lymphatic vessel draining it, and this lymphatic vessel is called the thoracic duct. Okay, and what will happen is the thoracic duct will ascend, so it will go up the L1 vertebrae, it will then go up all 12 of the thoracic vertebrae, and then it's going to drain into the subclavian vein on the left side. So let me talk about the great veins of the body. So basically, if we go up into your thorax, you basically have a bunch of large veins combining to make the biggest vein of them all, at least in the superior sense, okay, which is the superior vena cava. Okay, I don't actually know if the superior vena cava is bigger than the inferior vena cava, but it's a very big vein. Okay, so this is going to drain directly into the right atrium of the heart. Okay, and basically the two blood vessels that contribute to making the superior vena cava are the two brachiocephalic veins. So this one on this side is the left brachiocephalic vein, and remember, we're looking from the front, so we're always talking about patient's left. So if you imagine yourself rotated around and in this position, that would be your left side. So this is the left brachiocephalic vein, and then the other one on the other side is the right brachiocephalic vein. And then both of the brachiocephalic veins are then made out of two other veins combining to form one. Okay, like so. And the vein that is here is a vein that's coming down from the head, okay? And the one that I've pointed to is on the left side, so this is the left internal jugular vein. Okay, so left internal jugular vein, and that drains a lot of the structures of the head and neck. Okay, and then this other large vein here, and I'm running out of space terribly here, is known as the left subclavian vein. So this is the left subclavian vein, and that will be draining the left arm. Okay, right. So basically the thoracic duct will come up here, and basically it drains into the left subclavian vein just before the left subclavian vein combines with the left internal jugular vein to form the left brachiocephalic vein. So let me highlight it now in turquoise. So here is the thoracic duct, and it's joining, as you can see, the left subclavian vein just before it joins the left internal jugular vein to form the left brachiocephalic vein. Now, there is not two thoracic ducts. This is the one thoracic duct. It was in the midline, and then it sort of slants over onto one side and then joins the left subclavian vein. There is a smaller little lymphatic duct on the right side, but that doesn't drain the cisterna chyli. That just drains the thorax on the right side, known as the right lymphatic duct. Okay, so as far as bringing lymph from the intestine is concerned, the right lymphatic duct isn't relevant. This is the main duct, the thoracic duct, which is draining the cisterna chyli, and basically it tilts to one side, drains the left side of the thorax, whilst there is a separate lymphatic duct to drain the right side of the thorax, and then it empties into the left subclavian vein, and it's behind the brachiocephalic vein. So even though I've drawn it as though it looks as though it's in front of them, it should be behind all of them. So it goes behind the brachiocephalic vein here, and also behind the internal jugular vein, and then sort of latches on to the left subclavian vein. Okay, so basically the chylomicrons are going to come into the lacteals, they're going to drain into the cisterna chyli, they're going to go up the thoracic duct, and then they'll go into the blood via the thoracic duct, emptying into the left subclavian vein. Okay, right, so that's how we get the chylomicrons into the blood. So that's where we're going to finish the story. If you want to go any further, I'm going to have another video on lipoproteins and the transport of lipids around the body, which will be findable in my playlist on lipid metabolism. Okay, the one other thing that I want to mention is that Some free fatty acid molecules can go directly into the bloodstream, i.e. they don't need to go into chylomicrons and they don't need to go into the lacteals. Some of them can go directly from the enterocytes into the bloodstream. Okay, now basically to do this you need to be quite short. Okay, so you can't be like a really long chain carboxylic acid, for instance stearic acid. You need to be quite short. So you need to be either a short free fatty acid or at the very best a medium free fatty acid, okay? So only short and medium length free fatty acids can actually go directly into the blood. So what do I mean by short and medium length? Well, basically a short length free fatty acid would be something along the lines of a 4 or 5 carbon carboxylic acid, so for instance butanoic acid or pentanoic acid, whereas medium length might be something more around 10. Okay, so at a push, some of the smaller carboxylic acid molecules, which are rarer in food basically, the longer ones are more common, okay, but some of the shorter ones, if you do have them present within your food, they can sometimes just go directly into the blood, rather than having to go into the chylomicrons and then having to go this long diversion to get into the left subclavian vein via the thoracic duct. Okay, so that concludes our discussion of the digestion and absorption of fats.